Dear colleagues, first of all, I'd like to say that it's a great pleasure for me and a great honor for me to stand here. However, it's a bit undeserved because I substitute Professor Arseniev, who is in the hospital, and I wish him fastest recovery. It's also a great pleasure to give you an overview about the project I've been involved to since the very beginning of my scientific career, since 2004. And now I lead one of its branches. So the project is about the studies of functional association of membrane proteins of type 1. So first of all, I need to say that I represent here the Department of Structural Biology of our institute. And we have, if you can see, we have almost full capacity in studies in doing structural biology. We can get experimental structures of the proteins by NMR. We can do in silica experiments. We can do functional assays. So we try to use this setup and make a complex study of type 1 receptors. So first of all, a few words about the type 1 integral membrane proteins. If you listen to the lecture of Professor Schlesinger, he has spoke a lot about this. So type 1 proteins are the proteins which have only single transmembrane helix. These proteins have large extra and intercellular domains, so that prevents their crystallization as a full-length proteins. And they're also too mobile to be studied by cryo-electron microscopy at high resolution. The latest work I've seen reported the resolution of 27 angstroms, which is definitely not enough to draw any conclusions about the mechanisms of their actions. So these proteins regulate the cell cycle mostly. They are activated by protein ligands and are active as dimers or oligomers. And in 2004, when we started this project, there was not even a single structure of transmembrane domain available in the literature. So that was the point we were interested in. So these transmembrane helices. There are several views on the activation mechanisms of these proteins. There are two models proposed. The first model is called ligand-induced dimerization, and it implies that in the inactive state, the protein is a monomer, and the ligand binding causes the dimerization of the protein and subsequent activation cascade. However, there is another model which is supported by experimental data for some of the proteins, which states that in the inactive state, the protein is also a dimer, and the ligand binding causes some rearrangements in the dimer structure and subsequent activation. So the problem is that in case this mechanism is realistic, then we will need to find out two possible dimeric structures, the active one and inactive one. So our dreamboat, our ultimate goal, was to get active structure and inactive structure of the dimeric transmembrane domain at that time. And to do so, we use NMR spectroscopy as a major approach for structure determination. First of all, because we can look with NMR, we can look at both structure and dynamics of the protein. We can study thermodynamics and kinetics of the process. And we can use it to look at intrinsically disordered regions. And it is well known that type 1 proteins are quite rich with intrinsically disordered regions. However, we have a very limiting factor, very big limiting factor. It's the size of the object. It is limited to 50-70 kilodaltons. Otherwise, we have very high costs in our studies. So to start the project, we have to solve several issues, so several problems. First problem was technology of production and refolding of the proteins, and I will not focus on it in my talk. Second problem is need to proper membrane mimetic, because we cannot study the proteins in membranes. And we need to establish a protocol to determine the structure. And then the most important thing, we need to somehow interpret the structure and analyze the results. As I have said, our size of the object in NMR is limited. For this reason, we are not able to do studies of membrane proteins in liposomes or in membrane bilayers. So we have to use the membrane mimetic systems. We have plenty of them. We usually use three systems: micelles, bicelles, and lipid protein nanodiscs. Micelles are spherical particles of detergent. Bicelles contain a patch of lipid bilayer surrounded by detergents. And lipid protein nanodiscs contain the patch of lipid bilayer surrounded by the special rim protein or belt protein. It is also important to understand that membrane mimetic environment may affect the structure of the protein under investigation. So I gave here two examples. The first example is the example of glycophorin A. We've got structure in cells, in micelles, and even in X-ray, and we see no difference. However, on the bottom panel, you can see the structure of this beta-barrel protein, and it's completely different in 4 different membrane mimetics. We have structure in 2 kinds of micelles, in lipid protein nanodiscs, in crystals, and they're different. And I need to point out that this structure is native, and the structure in crystals is not native, for sure. So with that in mind, we need to say that we have to develop new kinds of membrane mimetics which properly mimic the bilayer and that do not unfold the soluble domains of the protein. For this reason, we made a lot of efforts in developing new membrane mimetic systems with new ring-forming detergents. We characterize their size and shape and the protein stability and native structure in this environment. And also here is the workflow of any NMR study of membrane protein, which is always started with very wide screening of membrane mimetic conditions, and which is always quite time-consuming and effort-consuming. So now when we have the membrane mimetic environment, we can start that the protocol is quite different for NMR studies to distinguish between intermolecular and intramolecular interactions in the protein. So we developed special approach which relied on the special scheme of isotope labeling of the proteins and of lipids. And this approach allowed us to directly detect the intermolecular interactions in homodimers of membrane domains. So once we have proposed this approach, we could manage to get the first structures. Here are the first structures which we solved in 2007 and 2008. Structures of the transmembrane domain of ERBB2, the famous anti-cancer target, and of the BNP3, it's a pro-apoptotic mitochondrial protein. So as far as we can get the structures, we need to analyze them. So we use MD simulations to study the adaptation of the structure to the lipid bilayer and to study the water accessibility and the interaction of the structure with the water. And another issue is that you need somehow to analyze the structures to get the some common conclusions. So there are several approaches. First approach is a motif approach, sequence motif. So you can search in the sequence some kind of determinants which are responsible for specific kind of interactions of membrane domains. Or you can study the surfaces. You can plot the map of the surface of its hydrophobic properties, and on this map you can indicate the interaction interface, and you can find out that it always or usually lies in the hydrophilic valley in the overall hydrophobic landscape of this protein. So apart from the structures, we are very much interested in the thermodynamics of the process of the protein-protein interactions in the membrane, because we need to somehow establish the structure-energy relationship, and we also need to somehow analyze the effects of mutations which are occurring in the transmembrane domains of the protein and cause different pathologies. So with this in mind, we developed a special approach which is based also on NMR. We follow the populations of states in NMR spectra and then analyze these populations taking into account the micellar character of our environment, and then we can get the free energies of dimerization. And moreover, if we study the temperature dependencies of these free energies, we can deconvolute these dependencies and find out the entropies and enthalpies of the dimerization, and which allowed us to find out that in many cases the dimerization of helices is entropy driven, which implies the very important contribution of lipids in the dimerization process. Furthermore, apart from the thermodynamics, we can study the static state kinetics. We can measure the on and off rates of dimerization and find out their temperature dependencies, plot the Arrhenius dependencies, etc., find the activation energy, which also showed the very, very important contribution of lipid bilayer, of lipids in the dimerization process. So now we have free energies and structure calculations. We've got a lot of structures. And recently, we managed to fulfill our ultimate dream and get alternative conformations for the same transmembrane segment of the same proteins. In the first case, we did it with mutating the specific residues which cause this mutation caused the spontaneous activation of the receptor, so we managed to get 3 different conformations of the transmembrane domain corresponding to 3 different functional states of the full-length receptor. Another example is the example of the Egfr proteins, where we managed to get the 2 different conformations in 2 different membrane-like environments. So we get one structure in micelles, another in bicells, and in this case, we get one structure in the presence of juxtamembrane regions and another structure in the absence of juxtamembrane regions. So now we have alternative states for 3 proteins. And however, if we have alternative states, we need to somehow assign them to the functional state of a receptor. So we need to say that this particular state is the active state, and this particular state is the inactive state. And this state is not realized in the full-size receptor. So we have several approaches for this. The first approach is scanning mutagenesis. However, unlike the common scanning mutagenesis, when you substitute all residues with alanines, we substitute our residues with glutamic acid and find out that glutamic acid at a certain position can increase the activation of the protein, or in some cases, 2 glutamic acids separated by 7 regions, 7 residues, which is the helical periodicity, can cause the ligand activation of the protein in the absence of ligand. So, and using this mapping, we can find out which conformations correspond to the active state of the receptor. Another approach is rational mutagenesis. So we can look in details onto these hydrophobicity maps of our helices and locate their potential regions which can be used for helix-helix interactions and mutate the residues on these potential surfaces. And analyzing the resulting data, we can assign this potential interface as active interface and this potential interface as an inactive interface. And the last slide here is about the nature things that we can find, because there are a lot of mutations in the nature which can cause some pathologies related to the overactivation of the proteins. So we can look at the potential sites for the hotspot mutation, gain of function mutations and loss of function mutations, and also map them on the interaction surfaces, on the hydrophobicity maps, and locate potential active and inactive interfaces. I'd like to finish with this part about the transmembrane helices and go slightly beyond the transmembrane domains. The problem with these proteins is that they are always studied with divide and conquer approaches. When you dissect the protein into several parts and then study these parts, get their structures, and try to assemble the full-size protein. So the problem of this approach can be illustrated using this wonderful slide, as I think so. You can imagine that you want to study how to play basketball, but you cannot make videos. You can only take pictures, and take pictures not of the whole human, but only of some parts of the human. So we can get a picture of a ball, of some complex between the ball and the upper part of the human, some kind of lower part of the human and medium part of the human. And what you get is some medium state of the human in the basketball playing, or some specific state. This can happen if you are lucky. If you are unlucky enough, you can get the inactive human. Or if you take the wrong conditions, you can even get a misfolded human, which this misfolded human will also play with the ball, but it would not be a basketball player. So we have the problem. We cannot correlate the states of the protein, of different domains of the protein in the functional relevant state. So what we suggest is to go beyond the transmembrane domain and study the junctions of transmembrane domain with other parts of the proteins. So we started with juxtamembrane regions, and we got several structures with juxtamembrane regions, which also allowed us to mutually correlate their state with the state of the transmembrane domains. And in some cases, we just find out that juxtamembrane regions, which were previously studied, and there were a lot of publications about them, appear to be the parts of protein transmembrane domains. And we can even go much further and get the structure of the protein in junction with the whole intracellular domain of the protein. So we did this for P75 receptor, and we measured the structure of the protein. We find the structure, the motions of the protein. We've got the fast regions and slow regions, and we concluded that the motions of the globular domain are uncoupled from the motions of transmembrane helix. And this allowed us to propose a new mechanism for the activation of this protein. And last here is that we even managed to get the structure of the full-size protein. This protein is called NR80D, and it's a co-receptor of previously shown P75. So we got the full-size protein, incorporated it in bicells, in our newly developed bicells, and made the special structure. It isn't published yet, but I hope that it will be published somehow. With this, I'd like to finish my major part of the talk and continue with the outcomes of our work, the global outcomes. So the first outcome is shown on this slide. So these are the special structures of transmembrane dimers which were obtained in the last 13 years. And this work was started by our lab. And I highlighted with the red rectangles structures which were obtained in by our group. So you can see that we've got a lot of information. We can get different states of the receptor. We can classify them. We can judge, we can think about what kind of states are realized in the inactive receptor, what kind of states are realized in the active receptor. And so we have a lot of data, we have energies, and this data itself is rather interesting. The next outcome is the activation mechanism. So looking at our structure, taking into account our data on the free energy measurement and our data on the kinetics and thermodynamics of the process, and the thing that we managed to obtain alternative structures in different membrane mimetics, we came out last year with this membrane-mediated signaling mechanism, which implies the active role of membrane in signal transfer across the plasma membrane. So according to our proposed mechanisms, extracellular domains of the receptors in the apostate somehow interact with the membrane, and this perturbs the lipid packing and allows the juxtamembrane regions to get into the membrane. However, when ligand binds, the extracellular domains are released from the membrane surface, and this results in the release in the lipid packing change around the protein, a release of the intracellular juxtamembrane regions and the active structure of kinase domain of this EGFR receptor. And the same thing we managed to do with the P75 neurotrophin receptor to propose a new mechanism which implied the active role of lipid bilayer in signal transduction process. The third outcome is the prediction software. So when we started our work, there was no prediction software and there was no structure. So as soon as we have a lot of structures, we can train the prediction software, and all these free prediction software packages were trained on the structures which were obtained in our lab. And last outcome is the models of full-size receptors. So as soon as we have transmembrane domains and crystal structures of the extracellular domains and intracellular domains, we can assemble the models of full-size receptor, and these models of full-size receptor can be considered as the view on the receptor activation process. So these two models, the model of ICR2 in the inactive state, the model of TLR4 were built in our lab using our structures of transmembrane domains. And this model was built in the lab of Natalia Yura, and in this wonderful paper in Cell in 2009. It was also based on our structure of transmembrane domain. So with this, I'd like to finish my talk and acknowledge our very crowded team, and especially acknowledge the Department of Bioengineering, which is headed by Akademician Kirpishnikov. They did a lot in this project to develop the production and refolding approach from the very beginning of its first phases. So, and our collaborators from Madrid and Willigen for their work, and funding agencies also for their support. Thank you for your attention.
Thank you, Professor Mineev. I think that doping-like problem will not involve so much in your studies. Yeah. Well, so this paper is open because he showed us some basket and other figures. So this paper is now open for discussion. Well, Mike, please, who has microphone in this audience? Yes, please. Who want to ask in this? Mr. Terich, you had I saw. So when people are thinking about, I may start, or you? You. Yeah, okay. So I have a question. Look, you showed us some model. I'm out of the field, but anyway, yesterday we have a wonderful lecture of Professor Saikulov from Philadelphia about the density of MHC on the membranes. He also used some models. And the biological, physiological effect depends very much on the density of the model, how much you put in this. So how you regulate this problem in your model, by physically and by modeling? Yeah, thank you.
Actually, what we do, we just get structures, and we have various oligomeric states, so we can make the proteins in different concentrations, if I understand correctly your question. So we can,
my question was,
big density,
there are some dependence when you put 1, 2, or 3 receptors together in the membrane, because the real situation may...
So of course there is difference. If we saturate our environment with protein, we get dimerization, then oligomerization, then further oligomerization. So we just select the state.
And you try to model also SH domains, so it's how it's, because the real contact is due to SH domain, which was discovered by Schlesinger, who was our plenary lecture. Yeah.
Right now, we're not doing this MHC complexes, but right now what we do, we try to express the proteins with the intracellular domain. So we transmembrane domain and full intracellular domain, and then study the interactions.
Okay, thank you. Thank you very much. Some other question from the audience? You have to ask something?
Only one very small one. Is there? Did you recognize any change on the bilayer, phospholipid bilayer, and any polymorphic change, or is it keeps the same as it is?
As we have shown that in the presence of the protein, the lipid bilayer is somehow freezed around the protein. So we see if you know about the states of a lipid bilayer, there is liquid state, gel state, and so-called the ordered state, liquid order state. So around the protein, we have the liquid order state of the lipid bilayer, and that explains why when it dimerizes, this coat of the ordered lipids is changed, and we got a huge entropic contribution to the dimerization process.
Thank you.
So, if no more questions, oh, you have questions. Please, you have chance.
My question is, do you suggest to have the lipids recycle the proteins, because there are some structures that can trap the lipid molecule?
Yeah, we observe the interactions between the protein and lipids. However, our proteins are quite small. We have only 2 transmembrane helices, so there is no place where it can squeeze into. And we see the almost uniform map of protein lipid contacts around the